design and which is excellent.
Did we have a sound check?
Yep. Just have a friend.
Should be.
Just to say your intro.
Lecture today or tomorrow.
Yeah.
Yeah.
Are you going to the conference?
No.
Yeah.
Are you doing? A bit far.
Too far, yeah.
Next year.
Next year is in France. Yeah.
Oh, you have.
Started. Morning, everyone. Thank you for coming. It's a great pleasure and honor to have Rokem, Dr. Ariel Rokem, visiting the KCNI today and giving this special lecture. So Ariel is a leading figure in the neuroinformatics and neuroimaging. He's known for many methodological and software contributions, in particular in diffusion imaging, with the live toolbox, among others. He's also known for a few training initiatives, including your academy. He's one of the authors of a reference book on data science in imaging. So Ariel graduated from a PhD in 2010 from the University of California at Berkeley. And then he moved on as a postdoc researcher at Berkeley first and then at Stanford. And he's now a research associate professor at the University of Washington in the department. So today Ariel is going to talk about neuroimaging data analysis in the age of AI. So thank you very much for visiting us. And looking forward to the talk.
Thank you. Thanks for the introduction and thanks for inviting me. I'm very excited to be here and excited to tell you about work that we've been doing and about maybe sort of vision of the future of neuroimaging data analysis in this new age. One way to frame this from a very recent paper that I just read last week is that we are entering a new paradigm of science. So these are the traditional four paradigms. The fourth paradigm that was introduced more recently as data-driven discovery, and maybe we are entering a new era. So starting from empirical science, just reporting the observations, theoretical science, putting in math and equations, that's maybe like a 19th century phenomenon, maybe a little earlier than that. Computational science, where we can run simulations. And then this fourth paradigm that was proposed when people started amassing large amounts of data, that we can learn from data and do data-driven discovery. And now maybe with the advent of AI systems, maybe we can start making autonomous discoveries. I have some doubts about that. But I think that if this will come about, it will be built brick by building methods that can do this. I don't think that we will reach some singularity and then all of a sudden, we will accelerate. I just don't believe that that's true. And I'll talk a little bit about both technical work that we're doing, but also at the end about socio-technical elements that I think are necessary in order for us to advance in that direction. And I'll start with data-driven discovery. Data-driven discovery in neuroscience has benefited from a transition that neuroscience underwent from more traditional lab-based work to a mixture of traditional lab-based work and observatories. Much like astronomy, we have brain observatories. These are large data sets that are shared between multiple groups, collected by large consortia. Paradigmatic example in human neuroscience is the Human Connectome project that collected really nice data that we're all using to this day, and even to this day is really state-of-the-art in terms of neuroimaging data collection from a very large collection of individuals, 1,200 individuals. Other data sets were more focused on specific kinds of applications like aging. This data set CAMCAN, the Cambridge Center for Aging Neuroscience, with 700 people throughout the lifespan. The Child Mind Institute's Healthy Brain Network, and the Adolescent Brain Cognitive Development that focuses on, particularly on mental health and particularly in adolescents and children. The Developing Human Connectome that focuses on very early life development. All of these with fairly large samples and very, very high quality data in some cases. And then, of course, the biggest gorilla, the big elephant in the room, which is the UK Biobank with its 100,000 individuals who will eventually have scans. This is a really unprecedented large sample with many different kinds of data. And so how do we go from data-driven to AI-driven science? What can we do with artificial intelligence in neuroimaging? I'll tell you a little bit about work that we've done to enable scalable research pipelines, to uncover complex relationships in data, and maybe to explain these relationships, to extract new kinds of information from existing measurements, and finally, to represent generalizable knowledge from images. And the focus of my scientific interest in asking all of these questions is the white matter. So the white matter is that large part inside of this image that contains the axons of neurons. The neurons are in the gray matter, and then their axons project long-range projections through the white matter. And it's white because the axons are covered with myelin, and that myelin provides the infrastructure for those neurons to message in ways that are fast, reliable, energetically favorable. And part of what's interesting about the myelin is that there is a program for myelin development. There are waves of development of myelin in fetal development, in childhood, in adolescence, and then in aging, there's sort of a fairly, what looks like a fairly a program for sort of senescence. But on the other hand, the white matter is affected by the environment as well. So for example, in this study where people learned how to juggle, you could measure reliable changes in the white matter in very particular locations. So studying the white matter is really a good basis for studying interactions between program for development and changes that happen with interactions with the environment as well. And we've studied lots of these things. We've studied early life development in a few papers, and we've looked at adolescence and childhood and impacts that development in adolescence and childhood has on reading abilities. We've looked at variance in cognitive abilities in adults and also mental health variations in adults. And then we've looked at the aging process in several of our studies. So I'm really interested in all of this and in a variety of ways that we can apply methods from image processing, from machine learning and so on to study multiple questions related to matter, development, aging, and individual differences. And the main method that I use in order to study this is data from diffusion-weighted MRI. Diffusion-weighted MRI relies on the fact that inside of these big bundles of axons that travel through the white matter, there's water, and that water tends to diffuse more readily along the length of these bundles than across their boundaries. And we can tune the magnet to be sensitive to diffusion, direction, and quantity in a location-specific manner. And that's called diffusion-weighted MRI. And from this data, we can infer properties of the tissue. For example, the mean diffusivity, you can see inside of the ventricles in this horizontal slice, you can see that there's a lot more diffusivity, and that's because there's much less tissue hindering the motion of water. You can see that in some locations in the white matter, there is higher what's called fractional anisotropy, meaning the water can diffuse more readily in some directions than in other directions, and that corresponds to the major white matter pathways. And then we can infer the principal diffusion direction, and that gives us a cue for computational tractography. So computational tractography or computational track tracing uses these inferences about the white matter in order to infer the trajectory of white matter connections. For example, if we zoom in on this box here and we model the diffusion every voxel in this image, we can put a seed in one location and then we can propagate what we call a streamline that follows the principal diffusion direction in every location. And if we put a seed in a different location, maybe it goes to other places. And there are multiple different nuances on these methods, but roughly speaking, that's what we do. And then we get a collection of streamlines throughout the brain, and we can analyze that in a variety of different ways. One way that I like to do that is to focus on large brain highways, tracks. These are known anatomical objects that have been validated using a variety of other methods, including in postmortem measurements. And we can find them inside of this collection of streamlines using a method that we call computational dissection. In computational dissection, we tell the algorithm or the computer, find me all the streamlines that go through this region of interest, for example, here the corpus callosum, and go to the front of the brain, to the frontal cortex, and that selects out a particular portion of the streamlines. And doing that, using a variety of different anatomical constraints, we can very reliably and robustly find these brain tracks in individuals in those large data sets that I described before. And we call that automated fiber quantification because once we find the track, we can look at the tissue properties along the length of the track. So for example, here's one tract in an individual, and we can collect the mean diffusivity along the length of that tract, and we can derive this one-dimensional track profile. So the x-axis here is the position along the track from posterior to anterior, and the y-axis is the mean diffusivity along the length of the tract. And we can do that similarly for a variety of different tissue properties. Here is the track profile for the fractional anisotropy for this same individual. And we've developed tools to do that. Actually, with a large community of collaborators, we've developed a whole ecosystem of tools. If you go to tractometry.org, you can find that whole ecosystem there in multiple different examples. The core of that ecosystem is a software tool that we call PyFQ that does all of this, the tractography, the delineation of the tracks, and then finding the tissue properties, and it gives you back the track profiles. But there are multiple ways in which you can use those track profiles to do statistics, to visualize. You can get your data from multiple different places, raw data, either in BIDS format or not in BIDS format, using a variety of pre-processing pipelines. And we've, over more than a decade now, we've developed this over multiple different studies with all of our collaborators and with funding from NIH and others to do this work. So we have these tools, and around the time that Anisha Keshavan came to work as a postdoc with me, we were really interested in looking at the Child Mind Institute's Healthy Brain Network. It's a large data set that will eventually collect something like 5,000 individuals. And together with Jason Yateman, who's interested particularly in reading in these individuals, we went to this data set and started looking at it, and we noticed that there is an issue. Children tend to wiggle around in the magnet, and you get data that's good in some cases, but in other cases, it's not that great. And we have a problem. MRI is sensitive to a variety of quality issues, and state-of-the-art QC requires visual inspection by experts. So how can we study this in a data set that has thousands of subjects? So we thought, well, maybe we can train an AI method to do this. Here's what we usually do in small-scale studies. You collect some data, you ask some neuroimaging experts to look at that, and then you have a data set. We did that in a subset of the data. But of course, this does not scale to thousands of subjects. So our solution is to train a computer vision model to do quality control for us. But how do we acquire enough training data? These computer vision models are really data hungry. And having experts look at enough data in order to even create the training data for computer vision model is extremely cumbersome. So we turned to the community and we developed a web app. So Anisha developed this web app, Brainder, in which you see an image of a brain, a single slice of the brain, and you can swipe left or right, depending on if you think that image is good or not. That's why it's called Brainder. And that worked really well. We advertised that in the community, and we got actually a lot of people to show up and do this. We heard about people doing this on the bus or in various in lectures and in various places. And you can get an average crowd rating for those images, and it's okay. It's not great. It's like an ROC area under the curve or something like 0.95. The next trick that we did was that we trained an algorithm to tell us who among this crowd is more consistent with the experts. So we found among the crowd of raters that some people were more like the experts and others were less like the experts. And we downweighted and upweighted accordingly, and we got even better at that. And then we can take that and use a deep learning algorithm in order to then train it on this much larger data set that we acquired from these crowds and get a very, very good algorithm with an area under the curve about 0.99 matched to the gold standard. This was the held out gold standard that the experts had rated originally. So we have an algorithm that really, really does well at this. And in the spirit of open source software, open science, we sort of opened this up and developed swipesforscience.org, which is this open source software that other people can then take and develop similar applications for any science that they'd like. We ourselves then, Adam Ritchie Halford, who was a postdoc with me, and in collaboration with Matt Cieslak and Ted Satterthwaite at Penn, developed fiber.dev, which focuses specifically on the diffusion MRI data. So what I showed you until now was on structural T1 weighted images. We developed then fiber.dev, and we figured out that we show a few slices and we show also these colored images that gives people a little bit more to work with. And we did that on kind of the full sample. We had at that point 2,747 samples from the Healthy Brain Network dataset, and we went through all kinds of curation, and then in the end we ended up with something like 2,000, just over 2,000 samples that were then rated by the neural network for us. Along the way, we developed some cool tools to scale a lot of the computation. In the way here, it requires a lot of computational power. So we developed some cool tools to scale all this on Amazon Web Services that Adam and I developed together. And in the end, we ended up with a model that is, approximately 0.95 on this thing, and went on and did a bunch of work that I won't tell you about today for lack of time with the Healthy Brain Network. Happy to tell you about that. One thing that I think is cool about this work is that the ABCD study and people in the ABCD, particularly Damien Fair, saw this work and said, hey, that's pretty neat, we should do that too. And in particular, in the follow-up study, the Healthy Brain and Child Development study that's currently ongoing, and actually ABCD starts at age 9 and follows these kids longitudinally. HBCD starts at birth and follows kids earlier on. So it's a follow-up study on a younger population. And they are now using, this is from Damien's talk at OHBM, I think last year, maybe the year before. They're using what they call brain swipes, which is swipes for science adapted to the kinds of things they want to do in HBCD. So they're able to take our tool. They honestly hardly even talk to us, but that's the magic of this approach, is you put something out there and people will use it and use it very effectively. I'm happy for it. And in our case, we got our track profiles with the quality control. One of the things that you really see nicely in these data is that the brain changes over time. So here, each bin is 3 years. The darkest are the youngest kids, about age 5, starting at about age 5, up to age 21 or so. And you see the brain as a whole really changes throughout childhood and adolescence pretty dramatically. It's even more dramatically visible. This was FA along multiple different tracks. This is mean diffusivity. So you see mean diffusivity goes in the opposite direction, decreases with age, and that's because the tissue becomes denser, more myelinated, water is more restricted. It's pretty remarkable. The question is, can we uncover some complex relationships, for example, predict the age of individuals? This is a common benchmark task in machine learning applied to neuroimaging data. The idea is if you have the true age of the individuals on the x-axis and the predicted age, you can use that then to sort of do things like look at an individual whose predicted age is a lot higher than their true age, or somebody who is maybe lagging behind, and make some inferences back from these kind of brain age models about them. But as a benchmark, it's really good because the signal is pretty robust and you can test different models on this. along the way, we kind of realized that what we're doing, trichotometry, is in fact feature engineering for machine learning. So it's a sophisticated way of engineering features for a machine learning algorithm downstream. And in our initial attempts, we thought, well, we have these features. So here now, in kind of a standard machine learning setting, Y is a vector with some property of the individuals, let's say their ages. So it's a vector with n individuals and their n ages. X is now a matrix that for each column has some tissue property, FA, MD, or something else for a particular position along a particular tract. And this has a lot of structure already embedded in it because the green columns here come from one tract and the orange come from a different tract and from another tract. And if we want to solve for beta, find the right beta weights, even in a linear case, we can take advantage of the fact that they're organized in this way. And we worked with Noah Simon, who's in biostatistics at UW, to develop a sparse group lasso approach. And this approach, you regularize using the lasso approach, both on the overall matrix, but also individually on tracks and across tracks as well, so that you get a solution that's tract sparse within tract sparse and overall sparse. You can take advantage of this structure. And that's something that we'll come back to in a little bit. I'll just show you some results from that. Here again is on the y-axis predicted age, on the x-axis the actual age. This is the HBN data set. I think at this point, excluding subjects that don't have great quality, we're something like 1,800 subjects. And of course, we developed open source software that applies this method. And we were pretty happy with that. But along the way, we started asking, could we do better with deep learning algorithms that don't require this kind of linear structure of the problem? And so Adam went to work and developed a whole series of convolutional neural networks and recurrent neural networks that we can run on the tracks. The idea here is that the tracks are contiguous in space, and so there's information in the spatial arrangement that we're not taking advantage when we're grouping them together and saying, oh, this is a group, but we're not saying these two are closer to each other than these two. In contrast, convolutional neural networks, recurrent neural networks do know about the spatial structure, and maybe they can take advantage of that. In addition, they can take advantage of some nonlinear structure that exists in there. And so what we're looking at here is we did an assessment on the x-axis here are the number of training samples. On the y-axis here is the accuracy and cross-validation. And so higher is better in this case. And what you can see, this PCR lasso is similar to our sparse group lasso approach. What you can see is that this improves the more subjects you put into the algorithm. That's kind of a known thing already. I'll just go to show you all of the results from all these different algorithms. What we found in kind of short is that the convolutional neural networks, for the most part, don't converge really by the time we've reached our maximal sample here. And maybe there's more potential for them to increase in performance. But really, the recurrent neural networks, in particular these bidirectional long short-term memory BLSTM algorithms, actually rise faster than the PCR lasso, so require less data and reach a higher asymptote. So there are some nonlinear relationships that can be taken advantage of in doing this task. But again, all of these algorithms are very data hungry. So the question is, what can we do with this approach? One place where we have applied this is in brain aging. And here I'm going to tell you about the case of glaucoma in particular. This is a collaboration with Cecilia Lee and Aaron Lee, who are ophthalmologists in the Department of Ophthalmology, University of Washington. John Cooper, who is a graduate student in my group, built this work. And the data set is the UK Biobank that I mentioned before. In the UK Biobank, we can find about 1,000 individuals who have glaucoma. And we found a large group of healthy controls to match to them. What you can already see here is kind of the width of the bar, and each bar here is the number of subjects, with that scale bar showing you what 100 subjects looks like. And then the y-axis here is the age, is that glaucoma tends to happen to older people. And that's sort of, we know that, of course, but this poses a problem to a machine learning setting where the age is confounded with the thing we're trying to classify. So what we did here is we matched for every individual with glaucoma an individual that does not have glaucoma, but has the same age, sex, and socioeconomic status. So we get this very balanced kind of sample. We had to drop a few subjects, about 10 subjects here, because we couldn't find a good match to match all the constraints here. But nevertheless, we could go and then look at these two samples. And in particular, we zoomed in on the optic radiations. The optic radiations are that big bundle that I showed you before that goes from the thalamus and is the primary input to the visual cortex that provides information about the visual field. And the hypothesis that we had is that if there's glaucoma on the front end in the disease of the retina, there might be some impact on downstream information transmission. So here is an optic radiation. In this case, it's actually from a kid in the HBN, and the track profile of FA. That's sort of what we zoomed in on here. And we looked at that. So here you can see that there are some differences. Here in orange are the controls. In blue are the glaucoma with error bars and 95% confidence intervals. For comparison, we have here the left corticospinal tract. That's the primary motor output. So it's in some way distinct from the visual system. And we find no difference there between these groups. Once you match for age, sex, socioeconomic status, the corticospinal tract looks remarkably similar. And this difference may be statistically significant, but the question we really want to ask is what is the practical significance? And that's because it's a lot of subjects. So in this case, a statistical null hypothesis test is a test of sample size, and the sample size is pretty large. So the question is, can we do something with that? Like, can we classify individuals? And we can do that. We can do that better in using the OR, the optic radiation, and we can do in these control tracks, the uncinate and the corticospinal tract, but it's really not that impressive. And this is using a linear method. And using the deep learning method, we can improve on that. So we get an improvement by using the convolutional, in this case, convolutional ResNet that operates on the track profile than we did from the linear method. So, and importantly, this model doesn't generalize to discrimination between younger and older subjects. We did this analysis because we thought maybe somebody could say, well, this is just an age detector. It just detects the tendency to be older, and maybe glaucoma is just an increased aging. So it's not the case that it discriminates between younger and older subjects. And it doesn't generalize to another retinal disease, age-related macular degeneration. But this model is a bit of a black box still, and we would like to sort of open up the black box. And so one approach that we've taken to opening up the black box is what's called SHAP analysis, Shapley additive explanations. SHAP, roughly speaking, for each subject, identifies the data features that most contribute to their classification. That's part of what makes it very appealing. Roughly speaking, you can think of it as like taking a subject and then looking at the model around their data and saying, okay, what drives the model around this position in the space of the features? And it gives some nice, interesting results. This is actually previous work that we did with Permita Mehta when she was a graduate student, also on glaucoma, also on the UK Biobank. So you find things like, for example, on the left side there, the x-axis is the intraocular pressure, the y-axis is the SHAP value. And if the SHAP value is high, that means for that person, the intraocular pressure indicates that that person has glaucoma. Here, glaucoma are indicated in orange, and healthy controls in green. What you can see is that, for example, intraocular pressure, which is almost the very definition of glaucoma, if it's very high, the algorithm thinks that person probably has glaucoma. It's very low, that goes negative, meaning I think pretty confident that person does not have glaucoma just based on this. And you see similar kinds of patterns with age, which also we know is a feature that's very informative for glaucoma. But you also find interesting things in places where you didn't necessarily expect, like in forced vital capacity, which is a lung capacity measurement. And it turns out that that's a very informative feature for people who have very low lung capacity. That is really indicative of the fact that they have capacity, that they have glaucoma. And this might be something about sort of some confounding factors. This is correlational, of course. It's very hard to say exactly what this means, but it gives a direction for future research at least. And we did something similar then with SHAP analysis with the track profiles. Here in this case, we're bringing the features into Z-score bins and looking at them. And one interesting thing to note here is that FA and MD both have, so here we have both for age and for glaucoma, FA and MD have these monotonically decreasing SHAP curves, but mean kurtosis, which is a different tissue property that indicates something about disorder in the tissue, or how organized the tissue is, that's actually nonlinear. And that might be part of the reason that the logistic regression doesn't do as well as the neural network in this case. And again, might be an interesting direction for future research. Okay. So moving on to another application, which is to extract new kinds of information from existing measurements. Here we're interested in brain aging again. Brain aging, as I've learned by collaborating with researchers at the UW on large studies where multiple individuals are measured over time, it's a really complicated multifactorial process. One of the ways that we as researchers of the white matter encounter this is by looking at what are called white matter hyperintensity. So these are flare images that are used in order to emphasize these white matter hyperintensities. And there's a question, what are these things? Radiologists don't always know they're looking at these things, but they do know that when the volume of this thing grows larger, that is correlated, strongly correlated with cognitive decline in aging. They sometimes appear right next to the ventricles. They sometimes appear deep in the white matter. And we'd like to establish the relationship between this and the tracks. So this is severe, this is relatively moderate. But there really is a larger sort of picture here, which is the relationship between white matter hyperintensities and the brain vascular system. So if you think about vascular risk factors like hypertension at the top, there's a cascade of different kinds of things that happen in the white matter that then eventually lead to white matter lesions. And this picture that is in this review by Zimmerman et al. Really drives it home. What's marked here are what are called the watershed regions. These are regions where the vasculature sort of meets, right? The veins and the arteries become thinner and thinner and meet each other in the white matter, and they seem to coincide with places that we often find white matter hyperintensities. This also from this paper from Ida Skola and Gottesman. This is kind of the model that we have right now, is that there are vasculature, and then when the vasculature withdraws, deteriorates with age, stiffens, changes its morphology, then this white matter damage happens. So we'd like to study the relationship between the white matter hyperintensities and the brain vascular system. But there's a challenge. Measurements of brain blood vessel morphology are not obtained in many research studies. Clinical data sets also often do not include these measurements. And but on the other hand, standard anatomical measurements that are acquired in many clinical, many big research studies and clinical studies contain some of this information. What do I mean contain some of the information? I'll show you in just a minute, but I'll tell you sort of about the method in which we uncover this. So the method that we use is generative deep learning networks. These generative deep learning methods take as input some image, and then they go through an encoding, some embedding, and then a decoding such that you get an output. And you can do this with brain data as well. You put in some brain data as input, and you look at the output of this encoding, decoding process. In past work, we used this in order to look at retinal imaging, again with Cecilia and Aaron, where we had images of the retina, standard optical coherence tomography images, and then we had these very fancy optical coherence tomography angiograms that show the blood vessels in the retina. And we trained a particular kind of encoding, decoding, this kind of generative network called the UNet, in order to match these to these and generate synthetic OCTA. I do believe the middle column is the synthetic and C is the true for this held out sample. And they're very similar, down to like really small details, providing a lot of utility because in most cases OCT is measured, but OCTA is not. And you can kind of look at the OCT images, the standard OCT images, you can find the features that the neural network is using. It's not magic here, it's finding some interesting features that are just very subtle in the images. And there's a similar thing happening in brain angiography as well. So here on the right is an MR angiogram, and those two, that's a T2 weighted and a T1 weighted, more standard anatomical measurements. And if you just zoom in on these brightnesses here that the MRA really highlights, which are large blood vessels, you can kind of see that they correspond to features of the T1 weighted and the T2 weighted. So the T1 weighted, the blood vessels are, as we call the title of the paper, are hidden in plain sight. They're there, but we usually really don't pull them out, and that's because we don't have the tools to do that. So what Asa Gilmore, who was a student in my lab, did was to train one of these UNet algorithms, a really sophisticated unit to take as input T1 weighted and T2 weighted, and then generate as output the blood vessel morphology. And we used another neural network called POSTA to take the magnetic resonance angiogram to generate the sort of ground truth blood vessel morphology. And that works pretty well. Here's one example, here in red, the synthetic, and in green, the ground truth. And that has a pretty good dice coefficient, not phenomenal, but pretty good. What's more important for us is that the features of the morphology, so here we have several different kinds of features, like number of branches, the total volume, the number of bifurcations, mean tortuosity, mean radius, things like that. They correlate highly between the synthetic blood vessel morphology that our algorithm generates and a ground truth. In this case, in this data set, we actually have a ground truth to compare to. In this case, it held out ground truth, of course. And so that's very encouraging. The question is, can we do something useful with that? So one thing that we did is we took it to another data set. This is the CAMCAN data set, the Cambridge Center for Aging Neuroscience. Here on the x-axis is the actual age of the individual. So you can see there's a large spread of ages in this sample. And what we did is we built an age prediction model based just on the features from the vessels. And that predicts pretty well in R squared of 0.41. But there's a problem. If you take cortical features, the cortical thickness in these anatomical images, you actually get a higher R squared. So the question is, do the vessel features contribute something different from what the cortical features contribute? And for that, we did both of them. So we built a model that has both, and indeed that model is better. And also we can show statistically that there's more explained variance in a model that has both vessel and cortical features relative to either of those. And that has relevance to cognition and health. So these features are associated with cognitive abilities, like the MMSE in older subjects, with blood pressure in subjects that are younger than 60, and that's above and beyond the age associations and above and beyond what you get just from a model that has just the cortical features in it. So here's an image of that. Here we have all the subjects. So the x-axis here is the age of the individuals. The y-axis are these different features. And what I've done here is I've filled in the circles for the individuals who have hypertension, and the curves are sort of averaged or kind of windowed averaged curves for hypertensive in red, normotensive in blue. And that shows you what is also a statistically significant result, that there is a difference in these features between normotensive and hypertensive, especially on the younger end. And really hot off the press, so we submitted this paper and they said, can you do something really useful with this, like Hell who has Alzheimer's? So we ran this on another data set, the OASIS 3 data set that has a pretty large sample, many of whom have an Alzheimer's diagnosis. And in fact, again, these features are associated with the Alzheimer's diagnosis above and beyond what you get just from the cortical features. Okay, so we've seen all these things. One of the problems that we still have is data-driven research like this is really data hungry. All the problems I've shown you so far required really large data sets. The smallest we ever looked at is like 700 subjects. But for many research problems, large data sets are prohibitively hard to acquire. In clinical applications, n equals 1. Integrating different data is really, really hard. Clinical data with imaging data. We've been struggling for years now with Ariel and Cecilia to even integrate between retinal imaging and brain imaging. Very, very hard. So what we are proposing to do what we're doing now and proposing to do in the future is to build models that represent some kind of generalizable knowledge from images. The approach that's been taken in many other applications is the building of what are called foundation models. Foundation models are models that are trained in a self-supervised way on masses and masses of data. And then once they're trained, they can be adapted to do a variety of specific tasks. In our case, we're building on the work of Meta AI researchers who developed this really, really impressive self-supervised vision transformer model called the Dino V2. And this model does things like identify different parts of an object and segment it from the background. This is a good video, right? It's cute. But also it can do things like, it can be retrained after it's been trained on a massive amount of images. Now that same model can be retrained to do both these kinds of things, but also, for example, depth estimation in an image, or in the same image, semantic segmentation on relatively small amounts of data and object detection and a whole host of other kinds of tasks. So the question is, can we build a foundation model for neuroimaging data? Here, what we did is we trained a model on 2D brain slices. The Dino V2 is a 2D model, operates on 2D images. So we trained it on 2D brain slices, and it looks like it can do a little bit of segmentation. What's more, I think, cool about this is that the same model that can be kind of trained to pull out different segments of the brain can be retrained to predict participant age. So this is an R squared of 0.6, working on the embeddings of this model, on the embedding layer of this model, and can predict in a different data set from what it was trained. It has never seen these data before in cross-validation, can predict participant age. So it's kind of tunable. So now we want to do this for real, and for real means in 3D. So currently, as in right now on a computer in some data cluster or data center on Amazon Web Services, training a 3D version on a large data set, more than 100,000 brain scans that we curated from multiple openly available data sets. We got some funding from the National Artificial Intelligence Research Resource Pilot near the NSF to get access to AWS's largest GPU computers, which are indeed very big. And we hope that this will be really useful. We hope we can do things like apply this model once it's learned about the data to data-starved image processing problems like neurodegenerative disease progression monitoring, prognosis, that we can develop new approaches to interpretable machine learning to answer questions about these data, like what are the neural characteristics of stage 2 Parkinson's disease. And that we can then take these and combine them with models of other related data. That's one of the things that the foundation models are used to do, to create multimodal models. So we're trying to build the little brick in the neuroimaging so that we can put that together with other bricks from other fields where there are huge advances being made. Okay, so I told you about artificial intelligence and neuroimaging and some of the work that we're doing. I think this is a really promising direction of research, of course. I think there'll be really cool things coming in the next, 10 years or so. And so I think we can do all of these things and much, much more. I'd like to end with maybe a little bit of a bummer. What is currently limiting progress? I've actually just wrote an editorial for Aperture Neuro called Human Brain Mapping at the Singularity. It's actually inspired by a paper by David Donahoe. And I think inspired by David Donahoe, my argument as well is that a lot of what is limiting us is socio-technical aspects of the work and not necessarily the technology, and that there is no singularities. We're going to have to build this stuff. So we're going to have to build robust and accessible computational infrastructure to train very large models. We've learned a lot about debugging distributed systems by working on this foundation model. It's been really interesting. We need to build open source software for neuro-oriented AI. We need the intellectual infrastructure for this kind of work. We need systems for curating, aggregating, annotating, sharing large data sets. We need the associated standards, no less the ethical frameworks. There are a lot of really open questions in all of these different things, particularly on the ethical side of like, how do we use this data in a way that preserves research subjects' rights and also the rights of patients. And then finally, we need to train neuroscientists how to do all of these things, which is a big challenge, one that I'm particularly interested in. Tristan mentioned that in the introduction. I think one of the challenges here is that neuroscientists have to learn a lot already. And now we're saying, well, learn all these additional things. It requires all the specialized knowledge about software engineering, machine learning, high performance computing, and many other things. And the question is, how can we teach all this? Our little contribution to that has been this project that, again, Tristan mentioned, NeuroAcademy, which we're doing for the 10th time this year. These are some pictures from NeuroAcademy. It's a combination of a summer school. In the first week, we teach people data science methods. And then in the second week, it's just a week-long hackathon where they apply these methods. Started that with Talia Arconi, and now run that with Noah Benson, my colleague at the University of Washington. And as mentioned, Tal and I wrote a book, Data Science for Neuroimaging, an introduction, based a little bit on our experience teaching. There's a kind of a book that you can purchase, but there's also a website that has all the content from that book, neuroimaging-data-science.org, and that we use to teach, and hopefully you can find useful as well. And with that, I'll thank members of my group and colleagues and collaborators and all kinds of sources of support, and I'd be happy to take any questions that you have.
before going to lunch. Thanks. Have some questions.
Boy, I think really inspiring work. It's very easy to see that how this is the future. In like, let's say, for example, foundation models or models where you're combining data from many different places, many different scanners, whatever, like, there's lots of different heterogeneous data. How do you avoid the model just learning noise or learning like, characteristics of how the different data sets were collected for something that generalizes, let's say to a new data set from a new place and new scanner?
Yes, I'll separate that into two things because I think there is real noise in the measurement that we need to think about. And then there is heterogeneity, like measurement idiosyncrasies, let's call them, right? That the measurement devices and what we call harmonization, right, between the different devices. Something that I glossed over a little bit in the achievement of Meta AI's team is that a huge part of that paper was actually automatically curating the data set that they used in order to train their algorithm from images on the internet. And that's because images on the internet contain a lot of things that you just don't want in your training set. They're either blurry, noisy, but they're just not useful. And so telling, oh, this image is really useful is a tricky problem in and of itself if you're collecting, in their case, billions of images. In our case, I think the fact that it comes from multiple instruments, actually, my hope is that the algorithm learns, oh, there are different instruments. Like I got a brain, it's got like a little brighter in the top and a little darker in the bottom. Sometimes it's the opposite way around. There's variation because of biological variation, but there's this instrumental thing as well. And the hope is also that it does learn about the noise in the data. One of the applications that we hope to use this for is to do denoising, right? So take this model and train it to, for example, take noised up data and clean it up into less noise data. The hope is that it learns something about the characteristics, the distribution of the noise. Now, this is all very hopeful and yet to be determined empirically, but I don't think, unless I'm missing something, I don't think we've baked in some fundamental flaw where the instrumental variability is going to necessarily sink the possibility that it can then learn, for example, about a new instrument. Right, and say, oh, I've seen different instruments. That's a different instrument over there in this new data set.
The same. Do you is your hope to just feed in the brain data without caring about the scatter parameters and voxel size, resolution, all of those things, because this is one of the big variabilities that we see in brain data, especially MRI. So is your hope is to have something that is much more generic that can, sweep over those differences and be able to recognize the features that you're looking for?
Yeah, we did some curation in the sense that, for example, we scraped all of OpenNeuro, and turns out OpenNeuro includes non-human brains as well. So we excluded the non-human brains out of that, but yes, we included in the modeling brains with different resolution of measurements. We do inside when we make the 3D patches, we sample the 3D patches to a certain resolution. That resolution is fixed, but it's inside of the model. Inputs can be whatever resolution, and the hope is that it will generalize. It'll say, oh, I've gotten this kind of interpolated image at like 1 millimeter isotropic resolution. It's been interpolated from, I don't know, in some cases voxels that are 1 millimeter by 1 millimeter by 7 millimeters, and that's also something that happens in this data. Yeah, again, we're taking our shot here. We got like the compute, so we said, well, and we have a time limit on that. Let's train this model on as much data as we can get our hands on and see what we learn from that process. And I think we're already learning a lot just on the technical side. I think we'll learn on those sides as well.
That's another question. Are you able to, so you mentioned Dino V2, which is the first time I've ever seen that. Like, are you able to, I think the term is transfer learning. Are you able to transfer kind of image-based learnings from other models that are trained? Let's say Meta's model trained on internet images like cats or whatever to brain images, or are you starting de novo with some completely untrained model and learning it on your 1,000 images? I guess a related question is 100,000 images enough?
I don't know, but that's what we can get our hands on right now. I think it would be very hard to go beyond 100,000 with datasets that are openly available and easy to download. If we could download the UK Biobank, we'd be closer to 200,000, but we are no longer allowed to download that data because they require you compute on their servers. So
You have a card that you can download like 40,000 images.
I don't think I can download those either at this point. Yeah, because I didn't download them before they put that restriction.
We have that.
I would like to have that. It's tricky though. It's not as easy as all that because we need to somehow, it needs to fit into your, it needs to fit into your data use agreement. But anyway, our hope is that 100, we don't know if 100,000, what 100,000 is enough to, but to your previous question, we were training de novo for two reasons. One is we did that with the 2D. We trained de novo and we could do something with it. And the other is there is no trained model on 3D images. There's no, we had to rewrite the code to do 3D blocks of images, and there just isn't a model that's been trained on 3D images before. So, that was cool.
Yeah. Thank you for your yeah. I have a question. Facing the same. If I'm worried, maybe even more. Because you mentioned, yeah.
Yeah, so the hope is that we bootstrap the process by doing one big model on as much data as we can, and that we can then translate that to smaller, for example, clinical data sets. We can take this model that's already learned knowledge, generalizable knowledge about images of brains, and we can then apply it, for example, to do things like Parkinson's disease staging. That's right now, I think, hard because it's limited. The data sizes, data sets that exist are just not big enough in order to do that. There's not large data sets with thousands of patients with Parkinson's at different stages. Instead, there are data sets that have, a few hundred stage. Those are kind of the largest data sets. So the hope is we'll learn what happens when you train a deep learning, my kind of models, when you train a deep learning neural network on some data, it learns two things. A, it learns kind of the front end filters, properties. It learns the features of the data, right? It does the feature engineering. So you automate the feature engineering. And you also need to train some kind of nonlinear relationship in the space of those features to do a particular task, like a classification task or some downstream task. So now we're doing, in this big model, we're training the feature engineering part, and these features are going to be hopefully then generalizable. We can take those features and apply them in many other problems. That's the premise of it.
We do have a comment from Dr. Felsky, and it's just Sri Joy is royal we. That's
That's about your access to UK Biobank, I think.
Yeah. So, you know,
like some other scientists are.
Yeah, so there's an interesting kind of sub-story there, which is the socio-technical story of how do you get a community interested in contributing their time. And there are two papers that we did on this. In the first paper, we put it up there, and Nisha is so charismatic, she talked to people, and they did it because it seemed cool and fun. The second paper, what happened was collaborating with Ted Satterthwaite, and he tweeted, come and do this, and you will be a co-author on the paper. And we got a lot of response to that, of course, and there are many co-authors on that paper. We ended up doing a little bit of a compromise in the sense that there is a consortium authorship in that paper, and that consortium are all the people who did and I think that's actually legitimately fair. Many of these people are not members of the community at large. They're members actually of our research community who saw this kind of visible person in the community saying, hey, come and help us, and you'll be a co-author with me. And they went with that. I think there needs to be some incentive, of course. Some of it is like you're bored and it's fun, the tasks that we do put in the community science apps, they have to be really small. You can't, we thought about like, hey, let's show them the whole volume. It's like, it's too long to kind of zoom through the whole volume. So we zoom through 3 slices instead. So they have to be really short so somebody can do that. And then we gamified it. There's the leaderboard. And you can get different levels, like your different animals when you do more and more of these ratings. As a complete side note, Jason Yateman, my collaborator, has now taken that and used similar technology in order to not really crowdsource, but put on the web the assessment of reading in children. Turns out you can use web apps to do assessments of reading in like 5-10 minutes in the child's home on a tablet that are correlated with the clinical best practices, like very highly correlated. So this has, of course, these kinds of technologies have many different applications.
Ask a lot about your foundation models
Yeah.
project. I'm very interested, both personally, and I think that's also a major direction for the future. Is it the end of, fine-tuned handcrafted features such as, everything that developed in FAQ, because we are going to rely on neural latent spaces that are going to, give us more generalizable features, maybe across teams?
I hope not, because I think there's there's a lot of room for like the anatomical knowledge, the biological knowledge to create features that are relevant and interesting, and also that produce more interesting scientific results in some sense, right? If you are able to say, this is the circuit, and this biophysical property of this circuit is the important thing, then that's more interesting. And I think it will be very hard challenge in these foundation models to pull out those kinds of knowledge. We're already challenged, even when we use track profiles in these neural networks, we're already challenged to interpret what they give us. So no, I think we'll still need sort of this kind of hypothesis-driven feature engineering, like the PyQ, like this, and also in order to understand like what the data gives us, to be able to interpret the results.
Thank you again.